Week 7 talks about mitosis and meiosis. So this is a list of the learning objectives that I'm going to cover. So part 1 talks about the cell cycle and cancer. So before we go into talking about the cell cycle, we need to do a little bit of a review of the structure of the cell and what chromosomes are. So we have a cell, and inside of the cell we have a nucleus. And the nucleus is responsible for storing our DNA. So there's DNA inside of our nucleus. Now for humans, we have 46 chromosomes. "23 come from our mom, and 23 come from our dad. So when our mom's egg fuses with our dad's sperm cell, they unite through the fertilization event, and that's what ends up giving us those 46 chromosomes. As individuals that have moms and dads, we have 2 copies of each different type of chromosome number. So we're considered diploid. So what does this mean? Well, our mom gave us chromosome 1, chromosome 2, all the way to chromosome 22, And then the last chromosome was a sex chromosome. And our mom, because our mom is XX, can only give us an X for the sex chromosome. These first 22 chromosomes are known as autosomes, or autosomal chromosomes. The last set is the sex chromosome. Our dad is XY. So avoiding the topic of gender issues and that type of stuff, strictly speaking, on a scientific perspective, what makes a man biologically male and what makes a female biologically female is the presence of X or Y chromosomes. If the individual has a Y chromosome, they will develop a penis and likely produce sperm cells to go on to be able to serve the male role in the reproductive process. If the individual does not have a Y chromosome, they will likely produce a uterus, and that uterus will be able to be used in order to house a developing baby. So, biologically, male is XY, female is XX. Now with dad is also going to give us chromosome 1, chromosome 2, and so forth, all the way down to chromosome 22. These first 22 chromosomes are still called autosomes. The last chromosome is going to be the sex chromosome. And since dad has both X and Y, dad can give X or Y. If dad gives the X combined with the mom's X, that baby is going to be female. If dad gives a Y in combination with mom's X, then that baby will be male. So this is how the chromosomes are laid out in people. And in different types of animals and organisms on Earth, they have different numbers of chromosomes, different arrangements. They even have different sex chromosomes and different ways that they determine sex, like temperature-dependent sex and the ability to change sexes in the middle of their life cycles and so forth, and take on both roles of being able to produce offspring and carry offspring. So focusing on humans for this class, this is how we get to our 46 chromosomes. We have 1 through 22, plus the 23rd one is a sex chromosome. Dad gives us chromosomes 1 through 22, plus the third one, or plus the 23rd one here. We get 23 from mom, 23 from dad. And that's how we get our 46. Chromosome 1 on mom is similar to chromosome 1 on dad. When we look at chromosome structure, we're going to learn about the different banding patterns and how karyotypes are different diagrams of chromosomes. You can see chromosomal abnormalities and different regions that you can highlight with different labeled stuff. But all that we'll talk about once we get into a little bit more detail on heredity and genetics. But if you look at chromosome 1 of mom, the same types of information are carried as chromosome 1 of dad. So, for example, if chromosome 1 on mom happened to carry information about eye color, chromosome 1 of dad would also carry information about eye color. But it doesn't have to be identical information. If mom has blue eyes, she's going to have the blue-eyed allele, which is a version of a gene. If dad has brown eyes, dad's going to have the brown-eyed version of the allele, or allele version of the gene. So together, these would produce offspring, and the offspring would either exhibit a dominant or recessive trait, which again we'll get into in a little bit more detail in heredity. But now that you have a foundational knowledge of how chromosomes work and where they come from, going back to the picture of this nucleus and the cell here, every cell has 46 chromosomes, with the exception of the sperm cells and the egg cells, which only carry 23, and that's what we'll talk about once we get to meiosis. So all of our cells in the body, or a majority of them, are diploid. They carry 2 copies of each chromosome. During a cell's life cycle, a cell is going to perform its normal growth and function, and then it's going to be stimulated to replicate. What's going to happen is the cell is going to grow really big and split in half. But when we split a cell in half, we don't want to have half the genetic material in each one of those cells. Because if you can imagine, let's say our cells had 20 chromosomes and our cells divided, well, now our cells only have 10 chromosomes. And let's say that those cells divided, well, now they're down to 5. And if those divided, they'd have like 2.5 chromosomes. So eventually there'd be like fractions of chromosomes. We can't have that happening. So what we do instead is every time we go through the cell cycle, we actually replicate our genetic material. So the cell grows really big, the number of chromosomes double, so that when the cell splits in half, both cells end up with equal quantities of chromosomes. And that's going to be the same quantity that the cell originally started with. This type of replication, known as mitosis, is the process of going from diploid to diploid. You start with 46, you replicate them, so now you have 92 chromosomes. And then when the cell divides in half, you go back to having 2 cells with 46 chromosomes. Why do cells need to divide ever? Well, let's say you get a paper cut on your skin. You need to be able to regenerate all of those cells that you cut through with that piece of paper. In order to replace those dead cells, the cells that are in place need to replicate. So you're going to have cell division to make up those cells or regrow the cells that have been damaged. Cells also have to replicate as part of creating a baby. So as soon as the sperm combines with the egg and they start to divide, all of that division, cell growth and differentiation, that all is going to develop a baby, which is going to eventually grow up to be an adult. That has to happen with mitosis. So we have to go through this cell cycle. So the cell cycle has two main phases: interphase, where the cell is not dividing, and interphase has a G1, S, G2 phase. And then we have mitosis, which is cell division. And then we go through the phases of prophase, metaphase, anaphase, telophase, and then following mitosis is cytokinesis. So this part, I've got separate slides to kind of go through the details of that, but we're going to go back a slide to talk about what happens during interphase. So if we look at this and we imagine that this is a clock and we're going in a clockwise direction, the first time period of a cell's life, and this can vary from a day to 20 days, but the first period of a cell's life is spent doing its normal function. So normal cellular function, normal growth. That phase is G1. So G1 is normal growth and function. It's just doing its normal thing. I mean, your liver cells are detoxifying the body, your stomach cells are absorbing nutrients, your endocrine cells are producing hormones. So it's just normal cell and function, or normal growth and function. When the cell gets triggered to divide, it enters into the S phase. And the S phase of interphase is when DNA is replicated. In other words, we make a copy of every single chromosome inside of the cell. So we started with 46 chromosomes. After the S phase of interphase, we have 92 chromosomes. Then we enter into G2, and G2 is the final growth for mitosis. This is when the cell is going to grow really large, because when the cell splits in half into two daughter cells, we want those two daughter cells to be sufficient in size to hold all 46 chromosomes. So G1, normal growth and function. S, we're replicating all the genetic material in the cell. And G2 is our final growth for replication or mitosis. Then we're going to enter into the phases of mitosis. When we start prophase, our cell has 92 chromosomes on the inside. What's going to happen during prophase is the chromosomes condense and become visible. Spindle fibers emerge from the centrosomes. So we have these centrioles. They're going to separate from each other, and we're going to start having spindle fibers come out. And spindle fibers are designed to attach to the chromosomes and pull them apart from each other. Basically, they're little fingers that can help separate the chromosomes. Then the nuclear envelope is going to break down and the nucleolus disappears. So when we talk about prophase and when I ask you about prophase on the exam, I'm going to ask you what happens. Prophase, the nuclear envelope disappears and the chromosomes condense. And then you have these spindle fibers that start to form from the centrioles. During metaphase, so in metaphase, these spindles are going to attach to the center of the chromosomes. The spindle is fully developed and the chromosomes align in the center of the cell. Imagine you have two children and they have a bucket of candy. Let's say they just sat through the 4th of July parade or went trick-or-treating and they got this huge bucket of candy. What's the easiest way to divide the candy amongst your two children? Well, you can dump it in the middle, and then with each hand you can pick up one piece of candy and separate it into opposite piles. And then with each hand you can grab one piece of candy in each hand and again separate it into different piles. That's what's happening when we divide our chromosomes into the two different cells. We have a cell, and the chromosomes are inside of the cell, and they're all replicated, and they're kind of hard to keep track of. So what we do during metaphase is, instead of having this random pile, we dump our chromosomes in the middle, so that each child cell will end up with equal amounts of chromosomes. And these cells are actually called daughter cells. So now a spindle will form from this cell to the center, and a spindle will form from this cell to the center. This one to the center, this one to the center, and so forth. And that's what happens during metaphase. All of the chromosomes align down the center of the cell. While this over here is really prometaphase or prophase. Now during anaphase, we're actually dividing the chromosomes so that each of the daughter cells ends up with equal amounts of chromosomes. So the spindles are going to pull those chromosomes toward opposite ends of the cell. So the chromosomes, as soon as they divide, they're known as sister chromatids. So we have anaphase here, and we have the spindle fiber again. And those chromosomes that were aligned in the middle split. So now we have half going to one side and half going to the other. And as soon as they split, we don't call them chromosomes anymore, we call them sister chromatids. So now you divided up the candy amongst your two children, and you need to somehow give ownership that this is your pile of candy and this is your pile of candy. So once we've separated the chromosomes, we need to ensure that boundaries form around each pile of chromosomes so that the cells are clear as to what chromosomes belong in what cell. That's where telophase comes into play. During telophase, the chromosomes kind of piled up in opposite ends of the cell, and new nuclear envelopes start to form. So by the end of telophase, nuclear envelope material surrounds each set of sister chromatids, or each set of chromosomes. So following telophase comes cytokinesis. Mitosis ends with just dividing the nuclei, but then you want to physically separate the cells from each other. Like, these guys have to actually separate from each other, otherwise you're not going to have true cell division. So during cytokinesis, in animal cells, a cleavage furrow is going to form. Basically, the cell is going to pinch in until the cells separate from each other. During cytokinesis in plant cells, a cell plate forms. So a new cell wall material is going to be built in between those neighboring cells. Either way, by the end of cytokinesis, you end with two diploid cells. Some cells in the body do not divide. So if we look at this cell cycle here, we just went through G1, S, G2, mitosis and cytokinesis. But cells like neurons don't make copies of themselves. If one of your brain cells dies, it's gone forever. Those cells slip into a phase called G0. So G0 is for non-dividing cells. But many of the cells in the body do divide, and they're going to complete the cell cycle. So now I'm going to talk a little bit about cancer. So what is cancer? Cancer is unregulated cell growth and division. What happens when cancer forms is a cell is triggered to divide. Now there's two different genes that are usually implicated in the development of cancer. Tumor suppressor genes, that's a U, are genes whose products regulate certain checkpoints in cell division. They basically prevent cells from dividing too much. If we mutate tumor suppressor genes, then we stop their function. And now what that kind of chaperone that makes sure that the cell's not dividing too much is gone. So the cell can start to increase their cell division. Another gene that's implicated in the development of cancer is called an oncogene. And that's a cancer-causing gene. So that's a gene that upregulates the cell cycle, and it's going to purposefully increase cell division. When we have mutations in these genes, or if we have dysregulation of this cell cycle that leads to the development of cancer, what happens is we increase the rate at which our cells divide. And that may not seem like a big deal, but let's go through the scenario. So if we have a cell that is supposed to divide every couple days, and now it's dividing every couple minutes. Imagine if you were a building designer and you were supposed to design and build buildings once a month. And now you just got new instructions that say you have to design and build these buildings at least 2 a day. Well, you're going to increase production and you're going to start doing this really fast, and it's going to look really sloppy. And the building's not going to be structurally sound, so it's not really going to function like it should. In the development of cancer, you have these cells that are made so quickly, and they're so immature that they don't even function properly. And that's usually not a big deal if it's something that's benign. If it stays in one spot, it's going to cause a localized tumor, but it's not going to spread, so your body can kind of handle that. Macrophages usually surround those types, so like these white blood cells surround those types of benign tumors, and they enclose them off so that they're not going to spread to other parts of the body. But let's say you have a metastatic cancer. Now this is a true cancer that metastasizes or spreads to other parts of the body. You have the development of these immature cells that don't look right and aren't functioning properly, and one of the cells in that bunch decides to fall off. And that cell gets into your bloodstream and migrates to a different area. Or it gets into the lymphatic system and gets pumped into the bloodstream and migrates to a different area. So now that cell lands in the new spot, and it's going to cause new tumor formation and stimulate the cells around it to also become cancerous. Now all those cells in that new area are also going to be made haphazardly, fast and sloppily, so they're not going to be functional. When that happens, now you've got a real problem. The initial tumor has spread to other parts of the body. Not only that, but tumors have this angiogenic property. In other words, they have blood vessels drawn to them, so they can promote the development of their own blood supply. And that's what you're seeing pictured here. You see this mass of tumor cells, and their chemicals have caused angiogenesis, or the production of blood vessels feeding nutrients right into the tumor. takes a lot of energy to build a tumor. And all of that energy is being stolen from other parts in the body by a direct line to your blood supply. If this is happening in one location, and then it happens in a secondary location or tertiary location and many other locations, a person can become seriously depleted of nutrients. This is why one of the major symptoms of cancer is weight loss. All of the nutrients that are supposed to be used for normal growth and function are now being funneled into the development of this tumor, which is non-functional. So it renders an organ non-functional, and it's eating all the nutrients of the body, which prevents, normal growth and function and will eventually kill a person. So cancer is bad. It's a dysregulation of the cell cycle. We know that cancer kills people, and now you have a little insight as to why.